This is fine, I guess. Yeah. Good afternoon, ladies and gentlemen. It's a pleasure to be here in St. Gallen. I look at cancer from a molecular biologist perspective, so I think in terms of pathways rather than in terms of morphology. And I think that's a general trend in the entire field, that morphological assessment has served us well. Of course, being Dutch, I am, of course, partially proud that the microscope was once a Dutch invention by Antoni van Leeuwenhoek. But we have moved on, and now it's time to also start considering the genome to look at patterns of gene expression and at mutations. And these two types of analysis will complement future diagnostics of cancer. Now, molecular analysis has brought us a lot of good things. It has brought us targeted therapies. But you can see from these Kaplan-Meier survival curves that treatments with targeted agents are sometimes resembling the Greek mythological tragedies, where initially it looks good, but in the end it ends bad. Because if you look at the curves for progression-free survival, for all of these targeted agents, they in the end converge, and in the end all patients progress and die. Of course, that should not come as a surprise to any one of you who is a medical oncologist or a medical doctor for that matter, because of course history has taught us that complex diseases like even tuberculosis and HIV/AIDS require combinations of drugs to fight resistance. So the answer to this sort of Greek drama of initial success followed by tragic progression requires, in all likelihood, combination therapies as well, which is possible because we have a large number of these targeted agents that can be combined in multiple ways. But here, I think, is where the problem really gets rather complex, because if you look at the number of targeted agents that we have in clinical development, then the number of even two-way combinations is nearly endless. If we have 1,000 cancer drugs in development, the two-way combinations is 499,000. If we were to test all in a phase 3 trial, we would need 500 million patients to test all the two-way combinations. And the answer to tuberculosis and AIDS was already a three-way combination. So with cancer, it will also be at least a three-way combination. So the number will be more people than we have on the planet that we need to test all the three-way combinations. So how are we going to deal with this? I'm going to show you how I think, from a genetic perspective, we should deal with it. I'm going to illustrate this with an example that I trust you're all familiar with, which was the example that the small fraction of BRAF mutant colon cancers do not appear to respond to BRAF inhibition as well as the BRAF mutant melanomas do. That, of course, is now, of course, a textbook material. My children in high school learned that a BRAF mutant melanoma should be treated with a selective BRAF inhibitor, and that works very well, as you see here. But why then does the same mutation, BRAF V600E, when present in the context of a colon cancer, yield such poor responses to vemurafenib? I thought that was, at least from my perspective, I can imagine that you find this frustrating, but as a molecular biologist, I find this interesting because there is, of course, some underlying biology behind this that we can understand and then learn from that. So I thought that the most logical explanation for this apparent lack of efficacy of BRAF inhibition in colon cancer was probably that there is a complex network of what we call feedback and crosstalk mechanisms in the cell. I draw that here for you schematically. Here we have a sort of hypothetical kinase cascade, kinase A, B, and C, that signal from the cell surface to the cell nucleus to maintain cell viability. And cancer cells depend on this cascade for viability. But oftentimes, when you inhibit such a cascade with a selective drug, let's say that kinase C is BRAF and we add vemurafenib, then the inhibition of kinase C leads to a signal to a second kinase that can be cell surface receptor as drawn here, that then becomes activated and now starts signaling also to maintain cell viability. So you see in this scheme that if this happens, then inhibition of BRAF, or in this case kinase C, is without effect, because even though the primary pathway was effectively blocked, the secondary pathway becomes automatically activated as a result of a hardwired feedback system that exists in the cell, and now that secondary pathway maintains viability. And obviously what we then would need to do is to also kill the feedback system that is now being activated when the first pathway is inhibited. Because if we were to inhibit both pathways simultaneously, as you see in the right diagram, then we would be able to maintain, to block the cell viability signal and induce cell death. Now, is this what is the explanation for what I just showed you, that the fact that BRAF inhibition in colon cancer is without effect? We know that there are 518 kinases in the human genome, and we can inhibit all of them, not all of them with selective drugs. We have drugs against maybe 20 or 30 of the kinases, but we geneticists, we can block any kinase in the human genome at will by silencing that gene by RNA interference. So we have RNA interference vectors for all 518 kinases. So if you look at this diagram, the rationale would be that BRAF inhibition in colon cancer would not be sufficient, but if we were to inhibit that kinase that is responsible for maintenance of that viability signal while BRAF is blocked, if we would block that kinase, the cells should die. So how does that translate into an experiment? Sort of schematically shown here. 96 well plates. We can inactivate each kinase in each well by using the RNA interference technology. We're not interested on the left-hand side, where we see that a couple of kinases, when we inhibit them, kill the cells even without the vemurafenib drug. Those are not interesting to us because they kill cells directly. That's what we call straight lethal. That is not what we're interested in. But if you look at the right panel, where we use the combination with drug, now we see that there's one kinase in the right upper corner where there is a second kinase that by itself is not lethal, but in combination with BRAF inhibition becomes lethal. That is the elusive kinase that becomes activated by BRAF inhibition, because if we now inhibit BRAF and that second kinase, the cells die. So can we find that kinase, that hypothetical kinase that is being reactivated when BRAF is inhibited? Of course, as a geneticist, I need cell models to study this in. Now, what was extremely fortunate is that what Scott Kopetz observed in the clinic, that BRAF mutant colon cancer does not respond to vemurafenib, is exactly what we find in the laboratory if we use BRAF mutant colon cancer cell lines. You see on the right-hand side a panel of colon cancer cell lines. The four top ones are BRAF mutant. They are treated with PLX4032. That's a preclinical name for vemurafenib. And you see that even at one micromolar concentration, they still grow happily. Whereas A375, which is a melanoma cell, are highly sensitive to the drug. And that mimics the clinical experience that BRAF inhibition in colon cancer is not a good idea, but BRAF inhibition in melanoma is a good idea. So that's excellent, because now we have a cell line model to study this intrinsic resistance of BRAF mutant colon cancer cells to vemurafenib. So I'll go briefly through the experiment. The experiment is not very relevant for you clinicians because you may not find this very interesting, but the outcome is. But I'll go through how it actually is done a little bit so that you have a sort of sense for how we do this kind of stuff. We have hairpin vectors that silence each of the 518 kinases. They are packed in a lentivirus that's to effectively deliver these silencing reagents into the cells of interest. The cells that we use here are BRAF mutant colon cancer cells. Remember, these are resistant to vemurafenib. We now infect these cells with each of these 518 kinase silencing reagents, and we treat these cells now for about 2 weeks, either in the presence of normal growth medium or with growth medium to which we have added vemurafenib. Now, you see here that each of these cells has a different color, and the color is artificial, obviously, but the color represents an individual, what we call molecular barcode. The molecular barcode is a unique sequence that we have added to the virus that tells us which gene is being silenced in which cell. So the molecular barcode is a sort of tag that tells us kinase A is inhibited in that cell, kinase B in that cell, kinase C in that cell. Each silencing reagent has a unique molecular barcode. We can fish out these barcodes relatively easily by PCR, and now we ask which barcodes are present in the left population that was grown in normal medium, and which barcodes are present in the right population. Now I told you that we're looking for a kinase that is lethal in the presence of vemurafenib, but not lethal in the absence of vemurafenib. So I'm looking for a barcode sequence that is missing in the presence of vemurafenib, but not in the absence. And if you look at this diagram here, you see that the blue barcode is present in the left side, but missing on the right side. And that missing means that the cells have died, because if the cells died, the barcode is gone. And that means that in this sort of schematic representation, the blue barcode is what we call synthetic lethal with vemurafenib. It kills in combination with vemurafenib. So can we identify that blue barcode? The answer is yes. We do deep sequencing because these barcodes, as I said, they're DNA sequences. So we count how often each of the sequences is present, and then we ask which one is missing. And we represent that in an MA plot. And I won't go in too much detail around the MA plot, but to our great surprise, we saw that the only gene that was killing BRAF mutant colon cancer cells in combination with vemurafenib was the EGFR, the epidermal growth factor receptor, which is, of course, if you think about it, bizarre, because you have all learned since 2008 that if you have a KRAS mutation, you should not inhibit EGFR with cetuximab or panitumumab upstream, because you have a downstream activation of the pathway, and it doesn't make sense to now inhibit the pathway upstream. Now here we have an identical, or at least almost identical situation, where the activation is BRAF, one step below KRAS, an activating oncogene, and now the genetic screen tells us that even though you have a downstream activation by BRAF, you still need to inhibit EGFR in addition, which is completely nonsensical if you think about it, right? This makes no sense whatsoever. So when I saw the results, I thought, God, what an utter failure, this experiment. But we did a very simple experiment in the lab because, of course, we have cetuximab and we have gefitinib. And you see here that in agreement with what you all believe to be true, that if you have a BRAF or KRAS mutation, that cetuximab or gefitinib has no use. And you see also here in this cell culture experiment that gefitinib or cetuximab alone do not inhibit the BRAF mutant colon cancer cells, and neither does vemurafenib, even at very high concentrations. But if we now combine two seemingly inactive drugs, we see that the combination suddenly becomes very powerful. Now this is, of course, a bizarre situation that two drugs that are each in isolation completely ineffective suddenly become so powerful in combination. But this is the concept of synthetic lethality that we were actually looking for. We were looking for two drugs that each alone do not work, but suddenly start working in combination because they each are individually required for survival signaling. If you block BRAF, you have EGFR signaling to maintain viability. If you block EGFR, you have BRAF left to maintain viability, but if you block both, you lose the viability signal and cell death results. So why is then EGFR activation required even though you have a downstream activation? The answer turns out to be that the moment you inhibit BRAF with a drug like vemurafenib, or as it's called here, PLX4032, you see that within 1 hour, you see that the epidermal growth factor receptor is activated by phosphorylation. And even though the BRAF MEK ERK pathway is blocked by vemurafenib, you see that the second important signaling pathway downstream of EGFR, the PI3 kinase AKT route, is highly activated as judged by the phospho-AKT. So even though we were successful in blocking the MEK ERK pathway, we have inadvertently now caused reactivation of EGFR through, and that led to activation of PI3 kinase AKT. If we now also block EGFR with either cetuximab or gefitinib, we see that we block both the phosphorylation of EGFR, but also the downstream AKT, PI3 kinase signaling, and now we have both the MEK ERK pathway silenced and the PI3 kinase AKT route silenced, and now the cells basically die from apoptosis. I won't go into much detail, but we have learned that the feedback loop that exists is that BRAF, MEK, ERK signals through a phosphatase called CDC25C that is required to now inactivate EGFR when signaling is activated. A classical negative feedback loop. Activation of the signal leads to a downstream activation that now shuts down the signal that limits the activity of EGFR. And what you do inadvertently when you treat BRAF mutant colon cancer patients with vemurafenib is that you short circuit that negative feedback loop. You can now no longer shut down EGFR signaling through the feedback loop. EGFR becomes hyperactive. The RAF-MEK-ERK pathway was effectively blocked by your drug, but the PI3 kinase AKT pathways goes berserk and starts signaling very highly, and that leads to proliferation and survival. And what you need to do is block this pathway as well. And now you see that your PI3 kinase AKT route is silenced and your MEK-ERK route is silenced, and that leads to success. Success means death in cancer treatment, and we see that indeed death is the result when we combine erlotinib or gefitinib with vemurafenib. We see the induction of cleaved PARP, which is a sign of apoptosis induction. We can show that in animal models of BRAF mutant colon cancer, we indeed see that cetuximab, or PLX as the chemical name for vemurafenib is ineffective as a monotherapy, and we see complete blockade of tumor growth in the combination of vemurafenib plus cetuximab in the animal model. And that has resulted in 3 clinical trials. I'll show you here one, which was the first one to get started. It is a trial with LGX-818, a BRAF inhibitor from Novartis, also known now as Oncoletter. In the dose escalation part, we have established a maximum tolerated dose for LGX plus cetuximab, or a 3-way combination with LGX, a selective PI3 kinase alpha inhibitor called BYL719, and cetuximab. We have now completed the dose escalation of stage 1 and stage 2 for these patients, and it's remarkable how well even the 3-way combination is tolerated. In fact, the clinicians tell me that the toxic effects of cetuximab on skin are completely gone in combination with LGX-818. So the 2-way and 3-way combinations are extremely well tolerated, and you'll see the first results presented of the clinical responses, which are I think are nothing short of remarkable, presented first at ASCO. So we're very positive about this study, and it's an example of a study that is based on a novel mechanistic insight. This is not a combination that is let's fool around and hope we get lucky with a combination. It's a combination that's rational, based on a crosstalk between a pathway. And pharma companies like that more and more, that it's based on rational combinations. So you saw that we published this finding in March in 2012. The trial opened actually in our institute in November 2012, and we had the first clinical responses within 1 year. So can we do better? I thought that the next big question was, why do we get such limited responses to KRAS, to MEK inhibition in KRAS mutant colon cancer? That is, of course, the bigger fish to fry here, clinically speaking, because there's more KRAS mutant colon cancers than BRAF mutant. MEK inhibition seems a good idea. It's a kinase downstream of the RAS signaling cascade. There's various drugs in clinical development. Selumetinib, AZ6244 is one of them. But if you look in animal models, none of the PDX models, and these are the best models, we have direct human into mouth xenograft for KRAS mutant tumors, we see hardly any responses. Very worrying, and I think the early results from the clinical trials are also not very positive for MEK inhibition in KRAS mutant tumors. So why is that? Well, if I look at my cell culture models, and the same is incidentally true for KRAS mutant non-small cell lung cancer, we see that the KRAS mutant non-small cell lung cancers nor the colorectal cancers, with the occasional exception, are very much resistant to MEK inhibition. Here we use selumetinib, the AZ6244. Very high concentrations are without effect. So you would say, Rene, but you just explained us what you need to do. If you inhibit the RAF-MEK-ERK pathway, you need to also inhibit EGFR. And so it makes sense to now use a combination of an EGFR inhibitor with a MEK inhibitor. So let's do that experiment here with gefitinib. No effect whatsoever. life is often more complex than we hope for, right? Because BRAF and KRAS are mutually exclusive in mutation in colon cancer. That says they're in a linear pathway, directly downstream from each other. Makes sense. That's why they are mutually exclusive. But if you have a BRAF mutation, the feedback system is EGFR, and if you're KRAS mutant, the feedback system is not EGFR, apparently, because EGFR blockade is not showing any synergy with MEK inhibition. So back to the drawing board. What do? We do a screen with all the kinases from scratch in KRAS mutant colon cancers and in KRAS mutant non-small cell lung cancers to see whether we can find now kinases that are lethal with MEK inhibition in KRAS mutant tumors. And the only kinase that we could identify here was ERBB3, a member of the family of EGFR, obviously, not an active kinase itself, but HER3 is different in that it forms active heterodimers with both EGFR and HER2. So it can form two active signaling complexes, EGFR HER3 and HER2 HER3. So gefitinib, I showed you, is not sufficient as an EGFR inhibitor. Pertuzumab as an HER2 inhibitor does not synergize very well with selumetinib. But if you use dual inhibitors that inhibit both EGFR and HER2, like afatinib or dacometinib, now we suddenly see the same type of synergy that we saw before in the case of BRAF and EGFR. So this data suggests that if you inhibit MEK, that you activate HER3, and HER3 makes two active signaling complexes, one with EGFR, one with HER2, and you need to inhibit both to now see synergy in growth inhibition. We know how it works. It's a different mechanism. It is a transcriptional induction of both ERBB2 and ERBB3. You see here a whole series of Western blots from different colon and lung cancer cell lines treated with selumetinib. And what you can basically see here, I don't know whether this is, I don't have an arrow here, but that's fine. Basically what you can see here is that both ERBB2 and ERBB3 are induced, and they are also activated as judged by phospho ERBB2 and ERBB3. So we see that we see activation as soon as we inhibit MEK with selumetinib, that ERBB2 and ERBB3 are upregulated. This is also at the RNA level, so this is a transcriptional process that's different from EGFR. That is a non-transcriptional process there, so the mechanisms are very different. And this is the result of loss of the c-MYC protein. I won't go into the molecular mechanism because, of course, you want to know the end result, maybe not so much the mechanism. MEK is a transcriptional repressor of ERBB2 and ERBB3, and if you now lose MEK, because if you inhibit MEK kinase, you destabilize MEK, the protein disappears, the repressor disappears, and the genes get activated. And when these genes get activated, we see very massive signaling, of course, again, through the MEK-HER pathway and through the PI3 kinase AKT pathway. So is there synergy between afatinib as a dual EGFR HER2 inhibition and MEK inhibition? In a short-term assay, you see here the induction of apoptosis. So now the curve is on the y-axis is induction of cell death. And you see that the purple line, which is the combination of selumetinib and afatinib, is the most effective in the induction of apoptosis as judged by caspase cleavage. So you see that also on the right upper panel of the Western blot, the combination of selumetinib and afatinib gives us the highest degree of cleaved PARP, which is a hallmark of apoptosis. And actually what we see is that proteins like phospho-BAD are completely disappeared. BAD is phosphorylated by ERK, and you see at the phosphor-ERK level that only the combination of afatinib and selumetinib completely silences the ERK kinase downstream of MEK. So basically what you do when you inhibit MEK, you try to inhibit ERK, which is the next kinase downstream from MEK, but that doesn't work because the upstream kinases, HER2 and EGFR, become reactivated, start signaling massively through MEK, and the drug concentration is not sufficient to completely block the MEK. So you get leak-through signaling as seen by the significant presence of still phosphor-ERK in the presence of a MEK inhibitor. Afatinib alone doesn't block it, but MEK plus afatinib now completely block phospho-ERK. There's no more phosphorylation of BAT, and BAT becomes active as a pro-apoptotic agent and kills the cells, as you see in the graph with the cleaved PARP. So that suggests that we should combine afatinib or a similar compound with a MEK inhibitor. In a xenograft model, we can see indeed that the only combination that gives us shrinkage is a combination of selumetinib and afatinib, or in this case trametinib as a MEK inhibitor and afatinib. So I think I've shown you that functional genetic can help us understand how to treat genotypes with combination therapies. BRAF in melanoma can be treated with BRAF and or MEK inhibitors, but in colon cancer you need to inhibit EGFR as well, because there is this feedback system that activates EGFR when we inhibit MEK or BRAF. But in KRAS mutant colon cancer, we need to inhibit both EGFR and HER2 because the feedback system is different and leads to this activation of HER3 that leads to activation of both kinases. So functional genetic screens can help us deliver precision medicine, smart combinations that are the result of insights in how signaling in cancer cells is wired. In fact, based on the work that I have shown to you, the last part is not even published yet. We now have 4 clinical trials ongoing based on these synthetic lethality screens. The first 3 are all based on the BRAF plus EGFR concept. They are either with Novartis drugs or with GICC drugs or Roche drugs. They all use the same BRAF plus EGFR inhibition, and all 3, I think, will report their first results at ASCO. The 4th one is based on the work that I just talked about, not even published yet, but the trial is already open, so we've done better now than we did with the first one. In the first paper, I had to convince Novartis to do the trial with the paper in my hand, and with the second situation, I had to convince the editor to publish my paper because the trial was already open. So we see we make progress in bringing our findings from the lab to the clinic to the extent that we can now start trials even before the papers get accepted. So NCT 203-9336 is a trial with dacometinib as a pan-HER inhibitor with a potent MEK inhibitor for all KRAS mutant tumors, whether they are lung, pancreas, or colon cancer. Finally, I think we should not forget that mutations do not tell us the entire story. Remember the seminal paper from Chuck Peru, where he identified intrinsic subtypes in breast cancer, the basal, the luminal, and the HER2 types that are now increasingly being used to stratify patients for different treatment regimens. I think it is only logical that colon cancer will go the same way. In the past year, you have seen publication of 6 or 7 different papers that identify intrinsic subtypes in colon cancer. We have published one where we see 3 distinct subtypes that have different mutation frequency. You see the A subtype being very high in BRAF mutations and MSI tumors, and the B and the C subtypes are very different in their biology from each other and from the A types. You also see that their prognosis is different. The worst case is the C types that we call mesenchymal tumors. They are very aggressive tumors, and they don't seem to respond to any therapy. Here you see the 3 types of colon cancer stratified according to their responses to chemotherapy, and you see that whereas the A and the B types in stage 3 colon cancer respond to chemotherapy, you see that the C types are fully resistant, something that we can also mimic in cell culture. If we activate TGF-beta signaling, we obliterate the chemotherapy response and the targeted therapy response, and we fear that activation of TGF-beta signaling lies at the heart of this subgroup of mesenchymal colorectal cancers. So I think stratification based on genotype and subtype is the way forward in oncology. Mutations alone are helpful, but beware, they do not tell the entire story. I think the most salient example is still the BRAF V600E in the context of melanoma or colon cancer, completely different story clinically, and it depends on understanding crosstalk between pathways to know how you should combine drugs for each individual patient in each individual setting. Functional genetic analysis, like I do in the lab, can help identify these combinations, and those together, I think, can deliver precision medicine for individual patients. And an over-reliance on mutations alone, I warn you, it will in the end will not do justice to what your patients will need. On that note, you see here some of the patients that are, people that are involved: Chong Sung, Liqing Wang, Sidong Huang, Anirudh Prahalat, Prashant Kumar, Wipawadi Granram. These are all very Dutch names. I'm sure you'll recognize them all. Roderick Beyersberg is the only foreigner. No, it's the other way around. Increasingly, of course, international work. Many clinical collaborators, Jan Schellens and Robert van Geel, I should mention, as the people that run the trials on BRAF and KRAS mutant tumors, and Alberto Bardelli as a trusted collaborator in a European project that we call Oncoletter. And on the right, you see some of the people that pay my bills. Thank you so much. Be happy to answer questions.